Welcome again to Jerusalem Biochemistry Hub, your go-to place for mastering biochemical concepts one step at a time. I'm still your host, Dr. Juliet, and I'm still on the subject of enzyme kinetics. Today, we should be looking at another major type of enzyme inhibition, which we call the non-competitive inhibition. In this inhibition, unlike the one we discussed previously, the competitive, here, inhibitors do not bind to the active site of an enzyme. Instead, they have their own site, which we call the allosteric site, where they bind to. So for that, we can have the inhibitor binding to the free enzyme or to the enzyme substrate complex. So they can either bind to the enzyme when it's free or after it is combined with the substrate. So this is inhibitor plus free enzyme. Enzyme substrate formed, an inhibitor attaching to that to give us an EIS. Substrate can even bind after the enzyme has bound because they are binding at different sites on the enzyme. In this kind of inhibition, we discover that Km is not affected. The affinity of the enzyme for its substrate is not affected because the inhibitor and the substrate binds at different locations. And also, what we discovered that the maximum velocity of the end of the reaction is reduced. So we have this still giving us our product and our free enzyme. Maximum velocity is reduced here, but Km is unaffected. And so it means that the Vmax is reduced by a factor, the same factor, 1 plus 1 over inhibitor dissociation constant. So if this is our Line-Wilber's equation, which we use for explaining types of inhibition, how is this equation affected by non-competitive inhibition? We're going to have our Km. This part remains the same. And then this part is affected. Vmax will be reduced by the factor of 1 plus 1 over inhibitor dissociation constant. And then our graph will also show that effect. This is 1 over V and 1 over S, remember? This should be a graph where there is no inhibition. This is 1 over V. And this is 1 minus 1 over K. No inhibition. So how will the plot of the presence of an inhibition look like? We're going to have something like this. It's going to be from the same place since this is not affected, but the Vmax is affected. So we can see the Vmax goes this direction. Presence of inhibitors, take the line on the Vmax upwards. Upwards. So this is no inhibition. And this is presence of inhibition. So Vmax is reduced by the activity of non-competitive inhibitors. An example of this kind of inhibition is what we see in cyanide poisoning. Cyanide is able to non-competitively inhibit the final enzyme of the electrotransfer chain, the cytochrome oxidase, thereby putting an end to chemoosmosis and also leading to cell death.